Welcome back, biology explorers. Today, we're diving into the fascinating world of chromosomes and the cell cycle. By the end of this lesson, you'll understand how chromosomes work, how they divide, and their role in health and disease. So grab your notebook and take some notes. Ready to learn? Let's go! Before a eukaryotic cell divides, its chromosomes condense. That means they coil up tightly and become visible under a microscope. Pretty cool, right? But what exactly are chromosomes? Chromo means colored, and somes mean bodies. So chromosomes are colored bodies. Every species has a characteristic number of chromosomes. Humans, for instance, have 46 chromosomes in their somatic body cells. A photograph of chromosomes arranged in pairs is called a karyotype. Each pair is called a homologous pair. Fun fact, in humans, one chromosome of each pair comes from the mother, and the other comes from the father. Now let's break down a chromosome structure. A chromosome consists of two identical chromatids held together by a centromere. These chromatids form after DNA replication, which happens during interphase of mitosis. The DNA is tightly coiled around proteins called histones. This keeps it compact so it can fit into the nucleus. And guess what? The protective caps at the ends of chromosomes are called telomeres. They act like the plastic tips of shoelaces. Cells divide in two ways. First, mitosis is for growth, repair, and asexual reproduction. It produces two genetically identical diploid cells. Second, meiosis is for making gametes, like sperm and eggs. These are haploid cells containing half the chromosomes of the parent cell. Let's review mitosis. The purpose: growth and repair. The number of divisions: 1. Daughter cells: 2 identical to the parent cell. And meiosis? It's all about variability. It produces 4 unique gametes to enable fertilization. Now let's dive into the cell cycle. The cell cycle includes interphase, nuclear division or mitosis, and cell division or cytokinesis. Movement between phases is triggered by cell signals called cyclins. The length of the cycle varies by organism, cell type, and environment. For example, root tip cells take about 20 hours, while human intestine epithelial cells take around 10 hours. When a cell needs to divide again, it receives a signal during G1 or gap phase 1. After the signal, the cell transitions into the S or synthesis phase. DNA replication occurs in the nucleus, forming identical sister chromatids during this phase, which is relatively short. There's a gap between the previous cell division and the S phase, and this is when G1 occurs. During the G1 phase, cells make RNA, enzymes, and other proteins required for growth. After the S phase, the G2 phase or the second gap phase occurs, where more cell growth happens, and new DNA is checked for errors and repaired if needed. Other preparations for cell division are also made, like producing tubulin protein for making microtubules during spindle formation in mitosis. When cell division goes wrong, it can lead to diseases like cancer. Cancer happens when mutations affect the genes controlling cell division. These mutations can create oncogenes, genes that trigger uncontrolled growth. Mutagens like radiation, chemicals, and certain viruses can cause these mutations. Not all tumors are the same. Benign tumors don't spread. Think of them like warts. Malignant tumors, on the other hand, invade other tissues, a process called metastasis. Stem cells are unspecialized cells that can become any type of cell. They're like the superheroes of biology. They're already being used to treat diseases like leukemia and arthritis. And who knows, future research might unlock even more uses. That's a wrap on today's journey through chromosomes and their features. Remember, every chromosome in your body is a tiny library of information, guiding how you grow and function. Don't forget to like, share, and subscribe for more biology adventures. Stay tuned for our next topic on mitosis.
See you next time.